I set a due date for this, right, of the 25th. And make sure that you post. This is one quick way to collapse and see who's posted so you know who you might want to reply to. Okay, so we talked about the 5 different classes of antibodies, the structure of an antibody, the different outcomes that happen when antibodies bind to antigens and which antibody classes can do those. So now we're going to actually talk about how it is we go about making an antibody, right? So last time, right, all about antibodies, but where do these come from, right? Why and how is it that we get stimulated to make them? And that is part of our adaptive immune response, right? So that was one of our poll questions even last time, right? Was to really key you guys in that antibodies are an adaptive immune response. So this is the most effective means for eliminating an invader. The problem with the adaptive immune response, it may take a week or more to build following the first exposure, right? The very first time you see that specific antigen, it takes time for the system to get online, right? A week or longer. Sometimes you don't have that kind of time. In that lag time that we refer to it as, your innate immune system components need to keep you alive, right? And keep it under control before adaptive comes online. So your phagocytic cells have to keep eating it, right? You have to wall it off into that area. You got to maybe even have complement MAC complex lysing those cells until we can get antibodies made to be really targeted and 100% more effective in eliminating the invader. The good news too with the adaptive immune response is it has a memory component. Whenever you've been exposed, you produce memory cells. These cells can very quickly be activated upon second exposure. So whenever you're exposed a second time, your response is that much faster, that much swifter. You're not waiting. You don't have that lag time of 7 to 14 days if you've seen it before. So vaccination really takes advantage of this memory component of our adaptive immune response. So for adaptive, you've got to have that first exposure to generate a response. So if we vaccinate you, that's an exposure, right? We're giving you the pathogen or a form of the pathogen or something that the pathogen produces like a toxin. We're getting your body primed and ready and to get it to produce memory cells. So that way, when you see it again, if you get naturally exposed to that pathogen, that virus, that bacteria, that toxin, your immune system has already seen it before. It says, oops, I recognize that guy, right? And swiftly responds. You're not waiting 7 to 14 days. You've already seen this one before. You know how to respond, and you respond. In this system, it is at a molecular level, as we saw, right? Those antibodies bind to little pieces on proteins even, or little pieces on a bacterial cell surface, or to one little spike on a virus. It is a very specific molecular interaction that happens. This system also has to be able to detect the difference between healthy self and dangerous entities. So we also often talk about knowing the difference between self and non-self. So this helps us obviously know that it's a pathogen if it's not us, and also when our cells are behaving badly, when they're potentially cancerous. We have ways of detecting this and eliminating them. Cancer is a big word, right? Related to lots of things, but probably the easiest way to think about it is your cells behaving badly, because that's truly what it is your cells have gone awry, and they're not doing their job, 2, potentially doing things they're not supposed to do. And any cell in our body can become cancerous. And so that's why there's so many different cancers and so many different treatments, right? And that word really just encompasses so many things for so many people. But in truth, the kernel of it all is it's our own cells, right? Doing what they're not supposed to do or not doing their job. Does that make sense to you guys? So we have a little bit of an ability to detect that. But of course, there's always things that slip through the system, right? There are things that slip through our immune system, right, that avoid detection, that use our own antibodies against us. That's why we still have illness and disease and even death due to illness and disease. So when we talk about adaptive, we've been talking about antibodies, right? They're one component of the adaptive immune response, and probably the one we knew the most about first, because they're very large proteins, right? We could be able to extract them from the blood and from mucus and things like that, and really study them early on. The cellular component was, we had to develop more advanced techniques to really know and understand what was going on between the cells of our immune system. So we sometimes break up, just like we break up adaptive and innate, but remember, these things work together, right? Just like antibodies can initiate the complement cascade. Complement cascade is an innate, you're born with system that's generalized to help protect you. But a product of your adaptive immune system can activate it. So they're interlinked. So even though we talk about humoral, which is sometimes referred to as antibody immunity, because this was that human element, right? These antibodies, they were this human element that we discovered. That's where it gets that name. But a lot of people, humoral doesn't mean much to lots of people, right? So sometimes they just say antibody immunity. Versus cellular immunity, where it's a cellular response, right? So we're going to be talking about what cells are responding as opposed to antibodies being the response. And this type of response is important, right? Because not all pathogens are the same. And so sometimes we need an antibody response. Sometimes we need a cellular response. Sometimes it's best if we have both systems responding, right? Both an antibody response and a cellular response. So we're going to watch a short video on the immune response. And let's see, hopefully I get all my... Okay, the other day somebody had that muted, but it's good, so let's see.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Okay, so he doesn't have the most entertaining voice, huh? Putting us to sleep this morning. What's the general name we give to B cells and T cells? Lymphocytes, right? They're lymphocytes. And one of the things they don't say in this animation is that even when T cells are activated, you produce memory cells for them as well, right? So all your lymphocytes, well, let me put it this way. All your B and T cell lymphocytes produce memory cells when they become activated and proliferate.
It said earlier in the video that the T cell had to recognize the antigen from the antigen presenting cell. How would it recognize it if it's never been exposed to?
That's a good question. So Dan asked a very good question. He asked, and I'll rewind to that part even. So the first step that they showed us was what's called antigen presentation, right? Where antigen presenting cell, and these are macrophages or dendritic cells, right, can engulf stuff and break it down by phagocytosis, and they're going to present it on their surface, right? And they use a particular class of molecule to do this that we're going to talk about. It's abbreviated MHC class 2. It stands for Major Histocompatibility Complex. And so as you can imagine, compatibility, what do we worry about? When do we have to try and figure out compatibility for somebody? Organ transplants, right, and blood typing, right? But more importantly for organ transplants, these are the molecules that we ran into problems with because the system couldn't communicate, right? When you take an organ from someone else's body, these receptors that present antigen are different from yours. Unless you have an identical twin, which I don't know about you, but I don't got one. So no spare parts person to help me out that's genetically identical to me, right? Unless someday they allow us to clone ourselves, which I don't expect that to happen, but you can see it in some sci-fi movies, right? If you like that kind of stuff like me. There is the potential, right? But genetically, we're different. And these genetic differences can create problems, especially with the immune system, when you've got to communicate, right? So it's like me walking up to one of you guys and trying to do a secret handshake that you and your best friend do. I don't know that handshake, right? I haven't learned it, right? So this is a very specific handshake that your cells do amongst each other to recognize each other. If they can't do that, then the system says, problem, right? Invader on board, right? And that's where and why people who get transplants have to be on immunosuppressant drugs for the rest of their lives, and why they're not going to go to festivals or work in daycares where you're going to be exposed to lots of people and potential pathogens, because you are purposely weakening your immune system to stop it from eliminating the organ that you got in order to live, right? Because unless your identical twin gave it to you, it's not a perfect match. And that's why, too, they're going to try and match it as closely as possible, why relatives are usually the best match, because you are relatively the same gene pool, right? But you're not identical. Right? You are different. So this communication, as Dan pointed out, one of the things that happened here is that a helper T cell came along and he said, well, it said it didn't it hadn't seen that pathogen before. How does it recognize it? Well, one of the really cool things about our T cells and our B cells is each one of them, when they're born, they're unique. this T cell receptor will only recognize a specific antigen. So you produce millions upon billions of T cells and B cells that have unique antigen receptors, right? So in the case of the T cell, it's just called the T cell receptor. But in case of the B cell, it's actually called an antibody, right, of the IgM class. That's why it had the antibody on its surface. So this is the time delay here, y'all, because if you don't have memory cells, if you haven't seen it before, you've got to find one of those billions upon billions of T cells, or several of them, because remember, as we talked about, there are different parts on an antigen. Even a protein has lots of different parts that we could actually recognize a whole bunch of different parts on that protein. But you've got to find, again, all those naive cells, all those naive T cells or B cells or both that recognize that antigen first. They're called naive because they've never seen the bad guy before, right? Once they see it, they're going to be activated. And we're going to produce memory cells, so if we see this again, this response is going to be faster. So as you can see, helper T cells, as their name implies, are going to help out other cells, right? And in this case, the macrophage is activating it. And remember, how do our cells communicate with each other? What did they release? Cytokines, right? And in this case, they actually named some specific ones, right? Interleukins 1 and 2, right? These are major players, right? So there's it's releasing cytokines. There's interleukin 1, abbreviated IL-1. There's IL-2, right? They're communicating with each other. And then you see over here, there's the two routes that we can go. We can go the cellular route, or we can go the B cell antibody route, right? Do we have to choose a route? No, sometimes we will go both ways, right? We will do both avenues of protection. We'll generate cytotoxic T cells that recognize that specific antigen, and we will generate antibodies that recognize that specific antigen, and maybe even multiple antibodies, excuse me, right? Maybe we produce 5 different antibodies that recognize the same bacteria or the same virus. But that's where sometimes this can get tricky, because antibodies, can they go inside of our cells? No, right? So if you're infected with a virus, do you want an antibody-only response? No, because if that virus is inside that cell, we're not going to be able to detect it. What's one system that's also a type of cytokine that's released will help us protect us from our innate immune response for viruses inside? Yeah, what are we going to release? What cytokines are we going to release when a cell is infected with a virus? It's going to tell neighboring cells that there's a virus nearby. Get prepared. This is an important one. Guys, don't wait till the last minute to study for this test. You will be very unhappy with your grade. Interferon. Interferons. Okay, interferons. So, antibodies can be helpful with virus infection, right? Because what can they do? When an antibody binds to a virus, what do we call that? That virus has been, or that toxin has been, yeah, but different term, neutralized, right? Because we're stopping the first thing that it has to do, which is attach to our cells, right? If it's coated in antibodies, can it attach to our cells? No. We've neutralized it. We've stopped it. But what happens when it's already got in the cell and say interferon isn't working? Do we want to be able to recognize that a cell is infected with the virus and kill it? Absolutely. So how are we going to do that? We're going to do that with cytotoxic T cells. All right. So you can see why with a virus, infection, you actually want both of these responses. You want antibodies because you want to maybe neutralize it before it even gets into the cell. But you also want protection for when it gets in the cell, right? If they get past the antibodies, we want to be able to eliminate those cells that are infected. Now for bacteria, what do you think would be the most effective response? A cellular or antibody?
Antibody.
Antibody, why? It binds to the bacteria and it would stop them from doing things like moving, right? Or attaching to us. It's also going to tag them for destruction by phagocytic cells. It's going to opsonize them, right? It may even activate what? That could lead to cell lysis of bacteria. Complement, right? Lots of things, right? Antibodies could do against bacteria. Do bacteria get inside of our cells? Do we need a cellular response? Some do. Some do, right? So although antibodies are really great for bacteria, and we probably pretty much all the time want an antibody response, we sometimes also need a cellular response against bacteria if they can get inside of our cells. So leprosy, or the PC name for it, Hansen's disease, is one of the most fascinating diseases to me immunologically. And I worked in research for leprosy when I first graduated from undergrad, before I went to graduate school. At the LSU Vet School is where the research center for Hansen's disease is, in Baton Rouge. Immunologically, pretty fascinating disease because for the most part, most people, your immune system can eliminate the Mycobacterial leprae. It's only they estimate about 12% of the population has a genetic defect where the bacteria are able to grow inside your macrophages and you can't kill it, right? So there's about 12% are susceptible. And then among the 12%, there is a range of response. Now think about this. This gets inside the macrophages and it can grow. So do we want a cellular response or an antibody response? Both, right? But we definitely need a cellular response, right? If it's inside of a cell, we want a cell response, right? We want cells to attack cells and destroy cells that are infected. Still with me? Okay. So there are some people that go all the way to that spectrum. They get what's referred to as tuberculoid leprosy, which means that they do a 100% cellular response where they produce cytotoxic T cells. Those T cells hunt down infected cells and eliminate them. And that's a pretty good response, right? Because we're going to kill the bacteria, right? Potentially. The problem is that this bacteria prefers the peripheral nerves, right? So these are your nerves, right, in the periphery. So in the arms, the feet, the ears, the nose. And this is why people get those deformities that they get from leprosy, right? Is from the damage. And it can cause extensive nerve damage to where they have loss of feeling. And so there was a patient story that was told to me when I worked there that he put his hand on a hot plate in another patient's room that was still on. He did not know that his hand was burning because he had no feeling in his hand until he smelt his flesh burning. Right, because he had no feeling, right, in his extremities. So like diabetic patients that'll have peripheral nerve damage, and actually some of the research they've done for Hansen's disease really helped out diabetic patients. They have to really make sure that if they get any wounds, that they check their body, because if they cut themselves, they don't even know they cut themselves. So even though we have antibiotic treatment because it is a bacterial infection, right, we have effective antibiotics against it. And we can essentially cure someone of leprosy, right, or we can eliminate the bacterial infection. If they had a very strong cellular response and they caused and this caused damage to the peripheral nerves, we can't repair this damage as of yet. So the people suffer with this loss and then any debilitation that may occur because of that loss of feeling. So that's one end of the spectrum, and those were the people that survived but yet had deformities, right, from the infection, and why, you know, they've been ostracized even since biblical times, and why they were literally sent to leper colonies, and in Baton Rouge was the last one in the United States, shut down in the, I think, early 90s. People from all over the world came here because we were the first place that developed the antibiotic treatment here in the United States, right outside of Baton Rouge in Carville, Louisiana, in St. Gabriel Parish. And there's a museum there. It's an excellent museum. And there's still a few elderly patients. There's one guy that's just hysterical from the Philippines. And again, he has loss of some of his fingers and toes to the disease. He doesn't obviously have it anymore, but he suffers for the debilitating effects that it had on him. Most of the patients at that time that were there were brought to a local hospital. And now it's treated on an outpatient basis. No one has to be, you know, ostracized and put in a colony of people for fear that you will get this disease. The other end of the spectrum, though, is a complete antibody response. Now, would this be the best response? If it's inside of your cells. No, those antibodies can't get inside of the cells. So only if, and this one, pretty much once it stays in, it just spreads cell to cell, kind of like tuberculosis does, within the macrophages and then within the nerve cells. So if you have an antibody-only response, the antibodies aren't doing you any good with this type of disease. And this is referred to as Lepromatous Leprosy. And so the bacteria will grow uncontrollably in these individuals, and they usually would succumb to the infection and die from leprosy. And they wouldn't have the deformities that you see in tuberculoid leprosy. But their spleen, their lymph nodes, their liver will enlarge with large numbers of the infection as it spreads even throughout the body. So for this one, it's probably the hardest to detect. But again, it can be treated with antibiotics. And then, of course, you have the other choices, right? We have people that fall right in the middle that produce antibody and cellular response, right? And so they're good because at least they're producing some of a cellular response. And then you have ones that are more towards the tuberculoid, but a little bit of antibodies. And then you have others that are more towards the lepromatous response, but a little bit of T-cell response. So we have about 5 different categories they would classify people with the infection with. And we would also do the same thing for the armadillos that they use to grow the bacteria, because they're the only known mammal other than us that get the infection. So people in Texas know and usually definitely in Louisiana, people know to avoid armadillos because they carry leprosy. And they'll get the deformities just like people do, but they'll also get the lepromatous form just like people do. And so we would actually test them to see which type of leprosy they would get if we infected them. So we would do a skin test, not all that different from the TB skin test, but in this case we would put the proteins associated with Mycobacteria leprae into their skin, and then we would biopsy it out and see if they had a cellular response or not. Under the microscope, you can see if cells infiltrated into the area or not. If they had a large cellular response, we didn't mess with them. If they had an antibody, mostly just an antibody response, then we would purposely infect them to grow the bacteria in large numbers in those organs to be able to harvest them and harvest the bacteria for study. Why would we do that? Why would we purposely infect armadillos to grow bacteria?
Scientific research for a cure?
Yeah, research for a cure, but how do we grow bacteria most of the time, y'all?
Oncolytes?
right? In media. This one grows inside of cells. You'd have to do cell culture to do bacterial culture. And they have not perfected those techniques to that level to be able to grow bacteria outside of a body. They cannot grow it in artificial culture because of the requirements it needs from the cells that it's associated with in the body. So one of the ways they actually produce more of it is in the foot pads, right? So actually in the pad of the foot of nude mice. Nude mice do not have a functioning thymus. So they don't have a cellular T cell response. They also live in bubbles, basically. They're like the bubble boy, right? They live in sterile cages, right, in the animal unit. I did not mess with them. I don't like mice. Not a big fan of them. And they were scary looking. They didn't have any hair. Nude, they mean nude, as in they have no hair. They're creepy looking. I thankfully was not tasked with working with them. I worked extensively with the armadillos, though, in infecting them, testing them, watching the progression, testing for the progression of the disease by measuring their antibody level in their blood as the disease progressed. And then when they were about tapped out, we would autopsy them for their organs to be able to utilize the bacteria from them for research purposes. So, fascinating, like I said, that spectrum, right? There are not too many diseases out there where people really tend to go one way or the other and have quite debilitating effects because of that. So immunologically, quite fascinating disease because we see all the choices there, right? We see people that go totally T-cell route. We see people that go completely antibody route, which unfortunately in this disease case is bad. And then you have ones that fall in the middle and then the peripheries. So there's one inaccuracy in this animation that I want to point out to you guys. And I probably need to point out to the publisher. I don't know if they would fix it. But, so here we have our cells, right? So all our cells can do antigen presentation. They use the MHC class 1 to do this. And because of that, they'll communicate, as you saw, with these cytotoxic T cells. And again, if that T cell recognizes that antigen that this cell is presenting, it's going to kill that cell, right? That's why we call them cytotoxic T cells. And they're going to eliminate that infected cell. B cells, when they become activated, and sometimes they need help from a helper T cell to be activated. Do you guys notice here on its surface that it has, here's its antibody, its B cell receptor, which can directly interact with antigen? Then later it would actually internalize this and present this antigen on its MHC class 2. This would allow it to communicate with this helper T cell that comes along. But when you see right here, look at, it's interacting here. It should be interacting with this molecule. Somebody goofed or skipped a step. The important thing is that interaction activates that B cell, and it's going to differentiate into a plasma cell. The reason why these guys are called plasma cells is because their cytoplasmic area increases dramatically because their rough endoplasmic reticulum quadruples in size for these cells. Now, it's the rough endoplasmic reticulum. What makes it rough? The ribosomes. And why do ribosomes make? Proteins. What are antibodies? Proteins. That's why, right? These cells enlarge. Why their rough endoplasmic reticulum increases. Because the rough ER is what makes secreted proteins, right? The ribosomes in your cytoplasm make proteins that stay within the cell. Proteins that leave the cell are produced by the rough ER. Even some of the ones that stay inside, but they stay inside vacuoles. They're not free-floating in the cytoplasm. Because lysozyme, right, and lysosomes, right, those vacuoles that we use in phagocytosis are actually produced by the rough ER, processed by the Golgi apparatus, right, and then they're in the cell. So they're going to produce, and as they said, right, some of them are going to be memory cells, and then we already know how awesome antibodies are, right? They can do things like you see here, opsonize, tag for destruction by phagocytic cells. They can neutralize this virus, right? Coat it in antibodies so it can't bind to our cells and it can't get in. So that's an overview, believe it or not, of what we're going to go into a little bit more detail. And I say a little bit more detail because there's a lot of details here that we're not going to go into. This isn't a graduate level immunology course, right? So we're going to keep it pretty basic compared to the amount of knowledge that we do actually have on this topic. But there's still a lot we don't know. Right? It's still a very active area of research, immunology is. So what do we mean when we say primary response, right? This is the first time that our body sees that specific antigen. The good news about the adaptive system, though, is it remembers, right? It produces memory cells. So if we encounter it again, right, secondary response takes over, and we rely on those memory cells. As I mentioned earlier, humoral immunity you can think of as being antibody immunity. And this is really important for extracellular antigens because remember, antibodies cannot get into our cells. So bacteria, right, an antibody response usually is extremely helpful. For toxins, it's going to neutralize them. Even for viruses, right, say in our bloodstream, before they get into our cells, this can be very beneficial to us. Right, when the virus is in the bloodstream or in the tissue fluids before it enters into the cell. Once something gets into a cell, like a virus has to do, right, it cannot replicate without using the cell's machinery, or an intracellular bacteria, we really need a cellular response, where we have cells that can detect cells that are infected and kill those infected cells. Both are very powerful and can actually damage our own body tissues if misdirected. So this system is highly regulated, right? We're going to talk about just a few of the interactions. There's lots of communication, lots of receptor, special handshake reactions that happen, interactions that happen to make sure that we want to mount this type of response, right? We want to be very careful. Verification is usually required. Right, we really want to make sure that the bad guy is in that building before we blow it up, right? So, before we start being destructive, let's make sure, right, that it's a bad guy. So it all starts with antigen presentation. Right? Very important part of this process. And dendritic cells play a major role in this. Even though macrophages can do this as well, dendritic cells are our main antigen presenting cells. So they're going to phagocytize stuff, and they're not going to just eat it up and keep eating in that area. They'll actually leave the area where the bacteria or the virus is, and they're going to share this information specifically with our T cells. Now it's going to have a choice depending on what type of pathogen it took in, whether it be a virus or a bacteria. It's going to present it in two different ways. It can present it on MHC class 1, and that will stimulate a cytotoxic T cell response. That will have it to tend to go down that road where we have killer T cells that will kill infected cells. Or it may additionally or only, right, depending on what's going on, what type of signaling it's receiving, what type of interactions it had as it acquired the antigen, it may also talk to or only talk to helper T cells. And helper T cells, as the name implies, are going to help out other cells. The main cells that they help are B cells. They can also help out macrophages become more super killers during an infection if they specifically recognize the antigen that was presented to them via the dendritic cell. B cells, remember, have their own receptor on them, and they can actually directly interact with antigen via their antibodies, their IgM class antibodies on their surface. And they themselves are antigen-presenting cells. They'll present to these helper T cells that have already been activated, and that communication with the B cell will help that B cell move forward in its activation. And it will also help make some decisions like, which class of antibodies should I make? Because a B cell without this interaction from a helper T cell, what's the first class of antibody and the only antibody that a fetus makes? And it's our largest antibody when it's secreted. IgM. B cells on their own just make IgM. But if a helper T cell comes along, that helper T cell, if it's in our mucus-associated lymphoid tissue, what do you think it's probably going to tell them to make? What class of antibody goes into our mucus? IgA. If this interaction is happening in one of our lymph nodes, which antibody is found throughout the inner part of our body, in our tissues, in our blood, in very high concentrations? IgG. G. Remember, E is reserved for Erica, right? So that one's going to happen with allergic response, right? But those of us that are programmed to go that route against certain things. So, this communication is really going to depend on the situation, right? So it's a pretty complex system that we're going to keep relatively simplistic. And then, of course, those plasma cells secrete antibodies that can bind to that specific antigen that they've encountered and help us eliminate that pathogen via the many different outcomes that can possibly happen for the different classes of antibodies. So B cells have their antibody, T cells have their T cell receptor. There's one major difference, B cells versus T cells, other than the fact that B cells make antibodies and T cells do not. B cells can directly interact with an antigen via their B cell receptor, the IgM class antibody it makes on its surface. T cells, on the other hand, cannot directly interact with antigen. Antigen must be presented to them by another cell. Depending on that cell, it's either going to be MHC class 1, which is on all of our cells, including our antigen-presenting cells, or it's going to be MHC class 2, which only our antigen-presenting cells can do. So that would be this molecule here that's holding on to the antigen, right? It's going to be MHC class 1 or 2, depending on what cell is presenting, right, and where the information came from. The T cell is going to recognize that specific antigen because of its unique T cell receptor. In addition, we've talked about two types of T cells, right? Helper T cells and cytotoxic T cells. They have different jobs, right? Helper T cells help out who? B cells and macrophages. T cells, on the other hand, that are cytotoxic go out there and kill other cells, right? Our own cells that may be infected. So how did we figure out this difference? One of the ways that scientists discovered this difference between these cells is that they had a different marker on their surface. It's called the CD marker. It stands for clusters of differentiation. Helper T cells have CD4 as their marker. This allows them to communicate with antigen-presenting cells expressing MHC class 2. So this is their secret handshake. Right? So how I remember this is I always remember 4H, right? 4 for CD4, H for helper T cell. Cytotoxic T cells, on the other hand, have CD8, and they communicate with MHC class 1. I also sing myself a little song for 4-H, right? So 4-H, 2, 4, 6, 8, who do we appreciate, right? Remember that one? So guess what? MHC class 2 goes with CD4, right? 2 and 4. 2, 4, 6, who do we appreciate, right? Whatever works, right? CD8, right? These are cytotoxic T cells. They talk to MHC class 1, right? There you could say our number one important cells, right? Because they are our defenders for all of our cells, right? Any one of our cells that gets infected, that cytotoxic T cell can recognize and destroy. And the number 8 sounds like to eat, right? So I always think about cytotoxic, CD8, they're eaters. They're going to go around and eat and destroy all the bad guys. Has worked for me for 15 years, those little stories. So hopefully they will work for you as well.
So the MHC receptors is where the antigen is?
Mm-hmm. Yep.
The CD...
The CD markers determine which MHC that cell can communicate with. So only helper T cells can communicate with MHC class 2, which is on only your antigen presenting cells. B cells, macrophages, dendritic cells. That's it. They're the only ones that do antigen presentation in that manner to talk to helper T cells. And so we're going to have a tendency, if it's helper T cell, remember, who does the helper T cell mainly talk to? You said it. What cells? B cells, right? And so what's going to be the response? What do B cells make? Antibodies. Other anti-word. Antibodies. Where if you have a cell that's infected, right, it's going to talk to cytotoxic CD8 cells, right? Those CD8s are what can talk to MHC class 1. And in this case, right, if a cytotoxic T cell communicates with the cell, it's infected, what's it going to do to that cell? It's going to destroy it. And we say that that's a cellular response, right? It's cell-to-cell response. So how and why and where does this all happen? Well, it kind of happens all throughout the body, right? But there is a system to help coordinate the efforts of this component of our immune system. And that is your lymphatic system. So you have several lymphatic vessels and organs that are very important in the functionality of your lymphatic cells, your lymphocytes. So lymphoid system is a collection of tissues and organs designed to bring T cells and B cells into contact with antigens. Right, so we can mount this response, right? As we saw, the B cells have got to see antigen, the T cells have got to have antigen presented to them. So one of the ways that we achieve this goal is having a vascular system that is not circular like our circulatory system, right? It does not circulate per se. Instead, it drains the pathogens from the areas where inflammation is taking place. Our primary organs, we wouldn't be able to mount this response without them. All your white blood cells are born in the same place in your body, right? It's referred to as your what? Where do all your blood cells come from? In your bone marrow, right? The stem cells in your bone marrow. So that's your one of your primary organs. But as it relates to your lymphocytes, they're named after the fact that one of them actually has to get further education because that cell really needs to know the difference between self and non-self and needs to learn that special handshake, right? Who does the special handshake? Which lymphocytes? So The T cells, right? Because they've got to talk to MHC class 1 and MHC class 2, right? They got to learn that, right? Where do they learn that in our body? In the thymus, right? And that's actually why they're called T cells, right? They're named after the fact that they must go to the thymus. Now this organ, which is right above your heart, underneath the breastbone, in your thoracic cavity, decreases in size. It atrophies after puberty. So there was a belief at one time that the organ stopped working. In fact, it continues to work, just not to the extent that it does before puberty. Why do you think it's working so hard when you're young? This is where it's learning the difference between self and non-self, right? And learning that special handshake for each cell that's generated. So when you're young, you're making a lot of naive, never seen an antigen before T cells, right? What happens as we age? The production slows down, because do we need that many naive cells? No, because what do we have more of at this point, hopefully because of exposure? Memory T cells, right? So stored in our body, we have memory cells. So we're not generating as many naive cells because hopefully we're having secondary, tertiary exposures to things we've already seen before. We don't need newly trained soldiers. We don't have to provide that huge space anymore. We can downsize. So after, and B cells, when they leave the bone marrow, they know their job. They travel to several sites throughout the body. Main concentrations are in your secondary lymphoid organs. And most of the homes where your B cells are going to go, and then your T cells after they leave the thymus, are going to be your lymph nodes, hence why they're named that, right? Because that's where the lymphocytes live. Very large collections in your spleen, your tonsils, your adenoids, which is a tonsil in the back of your nose, right? Your nasopharyngeal. I don't know why it's a plural term. Maybe they thought we had two at one time. You only have one tonsil in the back of your nose, the top of your throat. You have two palatine tonsils, though, in your throat. And then also your appendix is chocked full of lymphocytes. What is your spleen filter? Your blood. It is the only organ you can actually live without. If your spleen becomes damaged or ruptures because of disease or infection, they will remove it. If it's damaged in, say, a bad accident, and it's damaged to the point where it needs to be removed, they'll remove it. They will not give you a transplant, though, because you can actually live without it. Because its main function, again, is to filter your blood for pathogens. Your liver will take over this functionality when you lose your spleen and do a lot of the work that your spleen did. You are a little bit more susceptible, though, to bloodborne pathogens without a spleen. But you can live without that one organ. So when we have inflammation, right, when we have fluid leaking into our tissue because of possible invasion or tissue damage, especially with invasion, do you want that fluid to go right back into the blood? No, because it could contain bacteria or viruses, and once it goes into the blood, it gets a free ride on the superhighway to anywhere in your body to the next site where it may want to eat you, right? Do we want that? Definitely not, right? We don't want to give it a free ride. A red blood vessel at resting heart rate, y'all, can travel the entire length of your body in a minute. That's at resting heart rate of about 70 beats per minute. Right? So this is the superhighway. We are moving super fast. So how do we avoid it getting into the bloodstream? But we need that fluid back, right? We can't afford to just throw it away, right? We need to get that back into circulation. But we want to do that in a manner in which we're going to clean it. What's your question, Tyler? Or did I
say the spleen captures pathogens in blood?
Yes. So your lymphatic vessels actually start in your tissue, and they drain all that fluid out when we have inflammation. And all your lymphatic vessels combine into several trunks and then into 2 ducts, right? So everything combines. So it's just like the sewer system in our city, right? All the sewer lines lead to the waste disposal plant, right? Where it's going to be filtered before that water is dumped back into the river. How do we filter our lymph, that fluid that goes through our lymphatic vessels? And make sure there aren't any bad guys in it. And if they are, we're going to present them to the B cells and the T cells. What filters our lymph all throughout as it travels to the 2 ducts that dump it back into our subclavian veins? What are all these little green things? Lymph nodes. Lymph nodes. Your lymph nodes are crucial to filtering your lymph, that fluid that went into your tissues during inflammation, before that fluid goes back into circulation. So sometimes you'll end up with swollen lymph nodes, right? Especially we have a large collection of them in our neck region, right? And you'll go to the doctor, right? And he'll push on it and be like, oh yeah, glands are swollen, right? And you're like wanting to punch them because they're sore too and tender, right? You're like, yep, swollen and tender. Please stop now, right? That's actually a good sign. It means, right, that you've trapped them in the lymph nodes, whatever it is, and your immune system is responding, right? These cells are multiplying, hence the enlargement to the gland. If that gland is not tender, sometimes that's a sign of cancer, right? Because cancerous cells can get trapped in lymph nodes as well. But they're not going to cause the tenderness that we experience when it's infection. And in fact, if you have breast cancer, right, you have a central lymph node in your breast and then several throughout in the periphery. And when you have cancer, they'll go in and they'll take the breast tissue and they'll take lymph nodes and they'll test them right in the operating room for the presence of cancerous cells. And they're going to keep taking those lymph nodes until they don't find cancer anymore. And this is why some people who have had breast surgery because of cancer, they'll end up sometimes they'll have swelling problems in the shoulder armpit region because they've lost that network, right? It's been literally taken from them. So it has to find and reroute different ways to go to get drained out of that area. So it can kind of pool and stay in that area sometimes. And as you can imagine, our intestines are a large entry point, right? So your abdominal cavity is just loaded in lymph nodes, right, to be able to filter all that stuff before. And in fact, right, the fatty components of our food actually travel through the lymphatic vessels. Specialized lymphatic vessels referred to as the lacteals, right, in your small intestines. So in addition to areas where it's organized to an organ level, right, where it's encapsulated and you have different layers that you have in your lymph node and your spleen, you also have collections of cells that aren't organized to the organ level, but they are referred to as tissues. So you have mucus-associated lymphoid tissue, commonly abbreviated MALT, that's associated especially with your small intestines, right? This is a large entry point. So under the microscope, these are referred to as Pryor's patches, right? And they're large collections of B cells and T cells, and macrophages and dendritic cells, and even specialized M cells that purposely sample antigens from the gut to expose the immune system to them so that we can generate memory cells and potentially even antibodies. And of course, in this case, what class of antibody are we going to be producing in our mucus-associated lymphoid tissue? If we're putting antibodies into mucus, which class? IgA, right? IgA. And then your skin, right? We need protection for our skin. So again, you have large collections of white blood cells, your B cells and T cells and macrophages and dendritic cells associated with your skin. those collections are referred to as skin-associated lymphoid tissue, right? Commonly abbreviated SALT, which is kind of coincidental because our skin can be salty, right? So again, this is a blunt-ended system, right? The lymphatic vessels, the fluid that we draw up after inflammation into that system so it can be properly filtered and so we can carry in a safe way the pathogens to our presenting cells in the lymph nodes. That fluid is referred to as the lymph. And we already went over this, right? Different antigens and antibodies. I don't know why it's still in this PowerPoint. I guess I didn't take it out. Probably opened the wrong one. So, clonal selection theory. B cells and T cells are generated, right, in the bone marrow. T cells go to the thymus to get their additional education. They're able to recognize a functionally limitless variety of antigens, right? Each B cell, each T cell is unique when it's born. It only recognizes and corresponds to a single epitope, a little piece on an antigen. Although diversity of antigen recognition is random, which means we randomly make different B cell and T cell receptors. This is done at random. Probably why it takes so long, too, to try and find a match. Because we've got to search through, right? Hundreds and thousands and millions of different T cells and B cells to find the match. So the lymphocytes reside, they hang out once they're born, right, in those secondary lymphoid organs, in the spleen, in the lymph nodes, waiting for their dream antigen to come by. And again, it's their specific antigen receptor that's going to help recognize. Usually they're going to need confirmation, communication with another cell type, right? They can't activate per se themselves. And B cells, remember, are going to turn into plasma cells and actually secrete antibodies. Some of these guys will actually leave, like if they're been activated in a lymph node, they will actually migrate to the tissue. They'll travel to where the infection is. And they'll produce antibodies right on the scene. Once this invasion has been taken care of and there are no more antigens, there's no need to keep pumping out these antibodies, right? So those plasma cells will go through apoptosis. What does that mean? Programmed cell death. Does it elicit inflammation? No, one of the key things about apoptosis is there's no inflammation, right? Because this cell was programmed to die. Because we don't need it anymore. We don't want to feed it. We don't want to maintain it. Because if we see it again, who are we going to call up? Who's going to respond and turn into a plasma cell? The B what? B cell. More specific. So when we have this first exposure, right, and B cells are activated, some of them become plasma cells, other of them become ready for the second time this happens. Memory cells. Memory cells. Okay. So we don't need that plasma cell, right? We've got a memory cell that can turn into a plasma cell when activated. No reason to keep around that plasma cell. Costs too much money. So that's the last thing that we're going to look at is clonal selection. Oops.
The immune system can produce antibodies against millions of different antigens. These cells, which are constantly produced from lymphoid stem cells, are found in the blood and lymphoid tissues. Each B cell can synthesize only one of the millions of possible antibodies and displays this antibody on its surface. When an antigen meets a B cell having a surface antibody of the proper specificity, it complexes with the antibody. B cells that are not activated do not develop further. Stimulation of only the B cells that carry antibodies that react with antigen is referred to as clonal selection. Following antigen-antibody interaction, antigen-antibody complexes aggregate on the surface of the cell. This is called capping. The antigen-antibody complexes are then internalized. The activated B cell swells and begins to divide rapidly, producing a B cell clone. The B cells then differentiate into plasma cells and memory cells. Plasma cells produce the specific antibody that provoked its formation, whereas memory cells remain in circulation but do not produce antibodies. Memory cells can become activated by a later challenge by the same antigen. Many antibody responses require signals from T helper cells. The T helper cell is stimulated by an interaction with an antigen-presenting cell such as a macrophage. The macrophage ingests the antigen, digests it, and presents it on the cell surface via a class II MHC. The macrophage then activates a T helper cell that carries a T cell receptor capable of recognizing the antigen on the class II MHC of the macrophage. The B cell presents the antigen on its surface using class II MHC. The activated T helper cell interacts with the B cell and secretes chemical signals called interleukins to stimulate the B cell to differentiate into a plasma cell and produce antibodies.
And again, it's going to produce memory cells. Every time a T cell or a B cell is activated, even if it's the second time, it's going to produce more memory cells. So this one time it showed without the T cell helping, right? So we call that T-independent antigens. And you're only going to produce that first class of antibody that can be produced by the fetus, and it's the largest one, it's IgM. When you get help from a helper T cell, that's when what we call class switching will happen. Well, you'll go from IgM to maybe IgG, depending on the signaling and the location of this interaction, or IgA, or IgE, right, which I'm not too fond of, right? So there's lots of communication that happens, right, for these responses. Okay, so I will see you guys next week. One important note, although I think most of you guys don't travel to campus on Fridays, most of the faculty will be gone tomorrow. We're doing other duties. We have a conference in Baton Rouge. But they did send out an email saying that the student open computer lab, if you have computer work you need to get done, will be open tomorrow. It's normal Friday hours, which I think is 8 to 4. And there's no classes, of course, tomorrow.